very much, Professor Roman Efremov and Professor Vladimir Porayko. It's nice that this series of symposium is having its 29th symposium, and I'm very happy to present a talk which sort of reflect upon how pandemics will change the way we do things. So I try to talk about the pandemics were neither new or they were happening at a particular point of time. They were there from time immemorial, and then every time the pandemic strikes, there is some impact in every walk of life. And also in recent pandemic, we will see that there is a subtle change in the way in which we do science and technology. So then we try to introduce a topic where we try to do the drug discovery research or chemoinformatics and bioinformatics, keeping a particular disease in mind, a disease-specific web portal. And in this particular case, I would like to introduce you to the work that we have been carrying out to bring a disease-specific web portal called MPDS COVID-19. So when we try to talk about a disease-specific approach, I also would like to tell you that how this particular infectious disease, COVID, will probably demand different strategies compared to the conventional drug discovery strategies that are applied for other diseases. And we also try to talk about the Galaxy platform on which the NPDS is implemented. If you try to look at the epidemics and pandemics, they are time immemorial. So if you see about 5,000 years before, there were a house which is filled with skeletons, and that is related to a deadly epidemic. And about 2,500, the plague of Athens is one of the very well documented pandemic, which remains of Parthenon, one of the buildings of the Acropolis of Athens. And this city experienced a 5-year pandemic around 430 BC. And some people say that in some areas, no one is left to die. Almost every single person died. And in the 165 to 180 AD, when soldiers returned to the Roman Empire from campaigning, they brought back more than the spoils of the victory, because these people have brought a large number of infectious diseases such as smallpox, giving rise to the Antonine plague, which was really very, very disastrous. And there are a large number of plague of Cyprian, plague of Justinian, and then the Black Death. And we also have this famous or not so famous Spanish flu, which went and then killed before our COVID pandemic has struck the entire world. And there are some epidemics which are still continuing, like, for example, HIV or a Zika virus, and then from time to time, different viral breakouts are coming. So when we try to look at the various options, like prevention, cure, and management of a crisis are the most important things during a particular crisis, pandemic. Preventive management is adopting measures to reduce the impact of pandemics is very challenging, though planning is vital in mitigating impacts on socio-economical and environmental issues. So here, the focus will be on drug discovery or vaccine discovery, the supply chain, and also hospital management and policy decisions. There are different levels and layers of challenges that are there. And we strongly feel that whenever there is a pandemic which strikes, the best of the best science should come to the rescue of the people and the society to see how we can confront this particular disease. As COVID-19 spread, raising fears of a worldwide lockdown. Sorry, this is already there was a lockdown, and international organizations, scientists have been using this AI to track the epidemic in real time so as to be able to predict where the virus might appear next and develop an effective response. And basically, we can try to divide this task into identify who are at risk, what is the infection, what is the severity, and what is the outcome. And more importantly, when such a pandemic comes, we can try to get some new knowledge about what is the structure, what is the function of different proteins, and the protein folding, locating the isotope or epitope. And development of drugs are very important, and predicting the next epidemic and next pandemic is also very important. Then there are several lessons that one can learn from a pandemic like this. So when we try to look at what are the action items, the action items are in one group, and outcome is another group, and implementation is another group. There are quite a few activities that one can take, like diagnosis and surveillance, or quarantine, isolation, and then how do you prevent with masks, and communication, and developing the vaccines and antiviral strategies and antibiotics, and so on and so forth. And the morbidity and mortality and healthcare and other costs and social costs are also one of the outcomes. But the implementation requires a large number of different types of activities. And as you can see, there is a huge amount of data. When the data is high, the machine learning and artificial intelligence approaches will help. And it's very difficult to see that a particular disease is associated with a pandemic, and usually an infectious disease. And the disease cure may have a unique way, and a generic strategy of a drug development for all diseases has its limitations. So every particular disease will have a unique way of tackling that. So we have to look at the disease pathophysiology, and what are the different metabolic pathways, and what are the different interactions that are there, the gene-gene interactions, protein-protein interactions, what is the comorbidities like disease relationship. And there are several factors like immunity, inflammation, gut microbiota, and several other factors which are associated with this. And we wanted to introduce a concept like every disease will have its own way of getting manifested. So molecular property diagnostic suite is a disease-specific web portal which was conceived almost 20 years before, but we started working in 2011. And 2017, we tried to publish a disease-specific web portal, which is based on MPDS for tuberculosis. And the idea is this portal is aimed to gather, find, and discover all possible information on a particular disease from all possible resources, along with the CAT tools at one place. So afterwards, we also joined hands with Professor Poraiko's group. We try to extend this with the diabetes mellitus, and then recently we are trying to develop different modules, like a compound library module and a fragment library module, and several other activities are going on. And most importantly, we are trying to look at how different modules can be updated. And we also try to see that the disease-specific modules, which are required for a particular disease, are there, and they are about 30 to 40%. And disease-independent modules are about 60%. So, for example, if you take a literature or a target library and a gene library, that is mostly associated with a particular disease. And the data library is also associated with a particular disease, like all the information about the disease. Whereas if you look at the data processing, like file format conversion, or look at all the chemical space or the descriptor calculations or QSIR analysis, or if you look at active site analysis and pharmacophore-based modeling and virtual screening, docking. They are all disease-independent modules. And we have a large number of modules which are implemented in the new version of MPDS COVID, which we are going to present in today's talk. So this is the website which is accessible to everybody, and this particular website will provide the information that is available. In the left-hand side, you can see various MPDS modules and how to get data and draw molecules, and various other ways in which you can use this MPDS portal. And this essentially has the search tools where you can try to search and then browse and develop various databases in biological space and in the compound library, fragment library. And we also have an antiviral prediction machine learning tool, which are incorporated. And this particular material can be downloaded, and then some of the things we have put it in the GitHub. And the information that is available is put in the form of a manual, and this manual can also be downloaded. So we try to describe that MPDS is based on a Galaxy interface where we have the MPDS server, which provide different pathways and different generic variants and drug repurposing tools, and the modules which are based on the compound library and the descriptor calculations. They're all available, and this has the architecture which is within the MVC architecture with the Galaxy complementary components are there, and there are different tools which are obtained from the Galaxy toolshed, which I will try to describe that the Galaxy web portal also has several tools, and then we are trying to access these tools. The COVID-19 specific modules that are employed here are the protein information and various pathways that are there, the clinical information, the SARS-CoV-2 mutations, the polypharmacology approach and drug repurposing, which I will elaborate in the next slides. And we are also looking at the drug-drug interaction, the protein-protein interactions, the host protein information, the epidemiology data, evolution, the inhibitors database, and the literature. And we are also trying to look at what are the VOCs, variants of concern, and then other variants which are there, and how different proteins which are responsible in the virus, epidemiology, target information, and a lot of information which are very specific to that particular disease is being incorporated here. I just wanted to tell you that there are two things which are very, very popular now for most diseases, but for the pandemic, the drug repurposing appear to be probably the only option, because trying to develop a drug from the scratch is very, very difficult. So we can try to see that how the existing drugs, which might have a primary indication for a different disease, can be repurposed for that. And also, if a drug seems to be effective against the disease, the virus can undergo mutation. So it's important that we need to develop a drug which can attack more than one target at the same time with the polypharmacology. And then we have published recently some papers which have effectively utilized all the FDA-approved drugs with the 24 SARS-CoV-2 targets, and we try to look at the potential leads. And then there are a large number of drugs which were interacting simultaneously with 3, 4, 5, or 8, 9 targets. And there are drugs which were initially for CNS disorders or immunity, cardiology, infectious diseases, pain. These particular drugs have shown some indication against the SARS-CoV-2, and this could be a very useful information. And then we are working with our collaborators to see if they can come up with some ideas. And whenever such a thing is there, we may have to push our science, and we have in drug discovery the structure-based approach and analog-based approaches. However, recently, the protein-protein interactions appear to play a very important role And how do we decipher what are the different functions that these protein-protein interactions and assemblies are going to talk about? So host-virus interactions, virus-virus interactions play a very important role. For example, a large molecular assembly like this is there. We can try to get a lot of information by looking at the protein-protein dimers or multimers for viral entry. For example, the viral propagation, which we try to describe with this NSP12 and NSP7, which we have done with greater detail, or the RNA proofreading with NSP10 and 14. And we also have done something on the Hendra and Nipah virus, which might potentially be a threat that we will have to be focusing on it. And different methods can be used, like we have used extensively the MD simulations and mutation analysis, because we wanted to see the virus, what are the potential mutation sites, and how this mutation will affect the structure and function, and how will it affect the binding energy, and also, most importantly, what is the protein-protein interaction profiling that is there, and the residue-residue networking. So we can come up with a whole set of new types of analysis to understand from the viral entry, viral transcription, translocation, propagation, proofreading, and many, many other paths. And then that will probably give a lot of information about how we can really work with this. So I just wanted to make the long story short that there is a lot of scope in doing research. When a pandemic strikes, it will open some new doors. And artificial intelligence and machine learning is one approach which we have to use it, because the data that we produce by all these drug repurposing, polypharmacology, protein-protein interaction will be humongous. And it may be difficult for a normal conventional procedure to adopt all this data. And as we can see that now the rate at which the data is being produced in any area, especially during the pandemic, is very high, and AI/ML appear to be one of the approach. So why we have chosen Galaxy? It's an open source platform, because when a pandemic comes, it's not that we can see only from a commercial point of view, or the companies will play an important role. I think the whole community should come, and then Galaxy has an approach like this, a web service integrating a wealth of tools and resources, and lots of data is available. And this data is 100% open source. The source code is also available. The tools are available. It's very transparent, and it has an interactive environment, and it is accessible, reproducible, and this also adopts the FAIR principles. So we have been very actively working with this Galaxy platform, because we feel that this is a very good platform, because a script or a program written in for any given problem in CAD and related area can be added into MPDS Galaxy and made available as a tool. So for example, you can write a tool, you can write a small script, and this can be put here, and then we can try to put it in a local host, and then this particular tool is available. Once we put it, and then we can try to add it to the Galaxy toolshed, so that this particular program is available for anybody to use. So I just wanted to tell you that an approach based on disease-specific approach appear to be very important, and it is very relevant during a pandemic. Because mostly the pandemic will strike, and then before anybody can think, this might pose questions which might not have answers. So in such a situation, pandemics have a great potential to push disruptive research, and AI/ML have a great potential for the future preparedness. When Spanish flu strike, or even when HIV pandemic or something, there were other approaches, but I think we should make the best use of AI/ML during the pandemic by integrating molecular modeling and various other approaches. And MPDS COVID is an attempt to bring multifarious computational modeling approaches and data-driven approaches for the drug discovery research. And it's an open access initiative. And although it is not yet published, we can access the web portal, and we are looking forward to publishing this after this presentation. And this is possible by a large number of people working in this area, and people who are in red in the first paragraph are from the group, and they contributed to this area. And we are also thankful to the DBT Center for Excellence and Center for Excellence for Petroleum Research, DBT Bhairag Bionest, and CSIR. And I'm sorry to mention that there is also a DST RSF funding, Indo-Russian funding, that is there, where Professor Vladimir Paraiko and the group has very actively worked in this particular area. And I thank for all of you for giving me an opportunity, and will be very happy if there are some questions.
Thank you very much, Dr. Sastry. I see the first question in the chat from Vladimir Sulimov. Did you find any inhibitors of SARS-CoV-2 targets using your platform?
Using our platform, we have found a large number of inhibitors. Firstly, based on the drug repurposing. As I told, there were a large number of drugs which were found to have a new indication. And in addition to that, we also have screened the other databases, and we found some of the inhibitors and which are being tried. So yes, with this approach, we have found several inhibitors, and that are being tested.
Yeah, and Vladimir Sulimov continues his question. They were experimentally confirmed. Yes or not? Or so this is in progress?
Some of these things are experimentally confirmed independently by others, where when we are working on it, already some of the things are confirmed. And I have not seen a fully FDA-approved clinical trials, whereas there are things at different stages, and then there is a lot of data based on the animal experimentation and other things, and those data are available.
Thank you very much.
Thank you. So are there other questions? I have one more general, not philosophical, but a bit general question. In the title, you mentioned the term artificial intelligence. And how do you consider, in reality, the possibilities, the potentiality of these AI-based methods in drug discovery? Because often, I mean, people use this because it's just fashion, like kind of fashion. What is your opinion about real help of and powerful artificial intelligence?
This is a very, very important question. Actually, artificial intelligence and machine learning will give us a confidence to ask questions which we are unable to answer without that. And I was not expecting that I have this much time. Last year, we published a paper where we try to predict the failure of a candidate during the clinical trials. So this question you cannot have using a conventional approach. Okay. And when the volume of the data is very, very high, for example, you have a huge chemical space, and that some people say that this is more than 10 to the power of 60. When such high levels of data is there, you have to use some machine learning approaches. And artificial intelligence approach, especially based on the machine learning, text mining. I'm sure that you are already aware of some work that is done by Professor Paraiko's group on text mining. So the machine learning based on the text mining and other approaches appear to be absolutely important. And not only that, when we try to do a large number of molecular dynamic simulations, we get data, and this data can have a particular size of volume which could not be comprehended by simple statistics. So one has to use some advanced machine learning data mining tools, and which we have shown in several cases. In the last, I will be very happy if someone has some queries. I will be very happy to share some of the reprints that we have in the last couple of years. So to answer the question that artificial intelligence is indispensable, it's not just a fashion. This is absolutely important. This will give us a type of confidence to address questions. And earlier, we used to give a lot of importance for accuracy of a particular result. And artificial intelligence and machine learning tools will give a lot of confidence to come up with lots of probabilistic methods and give us a confidence to address a particular problem, like an epidemiology problem. And I wanted to also bring to the notice of this GPTs, the generative pre-trained transformers, the ChatGPT. ChatGPT appear to play a very important role. And some of the work that is in progress in our group, I thought that I will just introduce MPDS COVID. But ChatGPT has already made disruptive advances in almost all fields. And drug discovery is not an exception.
Okay. Thank you very much. We still have 3 minutes. And there is another question from Vladimir Paroikov. How quick your platform could be adopted to a new, currently unknown virus or other biogenic threats?
That's a very good and tricky question, because the platform is available and people can use, but when they start submitting a job, it will slow down. And the most important thing is it has to be installed locally. The Galaxy platform will come with a virtual machine, and when somebody wanted to use the resources like docking or like some other computationally intensive or demanding jobs, that cannot be run on the platform because they will have to run it locally. But whereas the search tools and other options in that, if they want to really look at the protein targets, and then if they want to do some search in our compound library and fragment library, that can be done very easily. However, if somebody wanted to use it very effectively, they will have to use it on a local machine, and then they can use the local resources, while the software is available for freely for anybody.